the stage 2 of the Krebs cycle, remember it's all about, obviously we're going to have the generation of our high energy electrons, but also to, in this context, just really regenerate the oxaloacetate so that we can start the whole cycle over again. And the first of the 4 enzymes that are involved in this is, it's called succinyl-CoA synthetase, but I'm going to call it the other name, succinate thiokinase. Regulation is kind of minor, and in terms of the mechanisms, what we're going to be doing is we're going to have a coupled reaction. What's so absurd to me, I'm not the greatest in the world at organic chemistry, but I can at least do a little bit of organic chemistry. But whenever I sit down and think about it, I understand that the hydrolysis of ATP, just if you look at the structure of ATP, seeing all those oxygen bonds to phosphate, all that close together just makes me uncomfortable, makes me nervous. But in terms of the thioester, I can't really explain, energetically speaking, why the hydrolysis of that yields actually more energy than ATP, but there's a lot of things I don't understand. In this context, though, succinate thiokinase is going to couple the hydrolysis and the cleavage of the thioester with substrate level phosphorylation to give us ATP or GTP, depending on the type of the cell. I wanted to make also about this in terms of the enzyme is that the phosphate group is actually transferred from one to the next via a histidine, which that shouldn't surprise you too much. Remember, histidine, we have that one nitrogen that has that lone pair or the nitrogen that's contributing to aromaticity, and it has that lone pair of electrons that could acts very reactively to act as a nucleophile at the phosphate group. That's not really so much important as much as you're understanding we're generating ATP out of that reaction. The next enzyme that I wanted to mention is succinate dehydrogenase. And one of the notes that I really want to make about this that I think makes it really unique is, the Krebs cycle is happening in the mitochondrial matrix, and I don't know whether it's the cristae or the actual inner membrane themselves, but succinate dehydrogenase is located physically very close to the inner membrane. Once I outline the mechanism, that'll probably make more sense later on. But what we're doing is we're basically taking succinate and we're going to convert it into fumarate, and obviously if we have dehydrogenase, we're going to be doing a redox reaction. The little bracket thingies there just means oxidation, but from this we can use this for something else. We're going to be using this for the conversion of FAD to FADH2. And this, I guess I'll highlight it in magenta, explains why it's located at the inner mitochondrial membrane. This guy goes straight or directly to the electron transport chain from there. As you know, FAD has it's less energetically speaking than NAD, sorry. In terms of making ATP, you want to have more NADH that's going to yield more per molecule than FAD will. And the reason that we use FAD instead of NAD, whereas in every other reaction involving an energy carrier we're using NAD, is because FAD, being in a lower energy state, that we're able to kind of conserve some of the delta G of our reaction, because the delta G of this reaction is really small. So yeah, the delta G of this reaction is really, really small. So that's why we use that. The next enzyme is called fumarase. The only thing I'm going to say about the mechanism for fumarase is that it's a hydration reaction, which hopefully you were able to probably observe that from the, the video itself, from the identity of the reactants and products. Last enzyme is malate dehydrogenase. And malate dehydrogenase is really a lot like succinate dehydrogenase in terms of the reaction that we're having. We're having the conversion of malate to oxaloacetate. I meant to draw that arrow in white, but oh well. And from the oxidation of this, obviously we can't have one without the other, we are going to be able to convert NAD plus into NADH. All right.